So, okay, I will talk about the regulatory requirements from the guidances. And I need to mention before, the guidances, they really focus just on a few transporters. And always to think about which tissue you want to treat with your drug. Maybe there's a special transporter which you really need to study. And that's why the EMA often say just the indicated transporter should be used. So they don't go so much in detail. So I want to start with a general overview. So most time the most drugs are applied orally. So when the patient takes a drug, it needs to be absorbed most time in the gastrointestinal tract. And there we have the enterocytes. So and we have a good model for the enterocytes, the Kappa-2 cell model. So the drug will be taken up there. Maybe it will directly metabolize apart. Maybe there will be efflux by PGP or BCRP directly back into the gastrointestinal system. So it means the bioavailability will be not maybe 100%. Then this part of drug which is available in the blood will come to the tissue which you want to treat. It will maybe metabolize in the liver, will maybe secreted into the bile, and that means it will go back into the gut, or it will be excreted in the renal system. So it means there's a distribution in our body. Then we have a metabolism, often in the liver, also in the tissue, maybe also in enterocytes, and we have 3 different ways of elimination. It means intestinal, bile, and kidney, and of course, metabolism is in the end also a kind of elimination. This is from the white paper from the International Transporter Consortium, showing in the general expression of the most famous or most important transporters. The figure which Roloff showed, it's much more detailed, much more transporters are shown there. All of them presented in the book. Not all of them, but not for all of them. It's known that there are drug-drug interactions, but it's for some of them indicated that there will be maybe a drug-drug interaction. So that's makes us feel very, very difficult, and it's still strong in development. So already mentioned by the FDA and EMA, there are the red labeled, or by the FDA, the red labeled transporters. And since last October, the new draft guidance from the FDA also included MAT-E1 and MAT-2K, also here for the hepatocytes. And what I don't like in this picture is, for me, I studied during my PhD a lot of OAT1, and it's also present in the blood-brain barrier. And for the treatment in the brain, you can apply inhibitors of OATs and you can reach higher concentrations. I just want to mention it. So therefore, think about which transporter do you really need to study for the tissue which you want to treat. What is the goal? What for the guidances for regulatory compliance? It's of course the pathway identification. So which way takes a drug after uptake? So the pharmacokinetic of the drug. What do the drug with our body? Is there a hepatic or renal accumulation if the compound is toxic? What's about the excretion also? And also transporters can be also a target. That means, for example, BCP, PGP, or MDL1 or efflux transporters in general. It's about also kind of safety. Also delivery. There are several different transporters. For example, for peptides, peptide-1. That means if your drug is a peptide-like drug, then this transporter, of course, needs to be studied in detail. Also for pharmacological reasons, maybe your you really have a target, which is a transporter. So you want to block the glucose-like transporter to or U at1. Then, of course, you also need to study this transporter. They are not mentioned in the regulatory, so that's why I mentioned them here. Okay, why should transporters be studied? There are two topics mentioned by, for example, by the EMA. They wrote it pretty nice. Each drug needs to be studied as substrate, as an inhibitor. And when should it be studied? It should be as early as possible. So, of course, for example, it's better to say it like this, the inhibition studies, they will be performed in the very early stages, most time almost in the discovery stage of the most drugs. And it needs to be really known, the inhibitory capacity before phase 3, before clinical phase 3, because it's the biggest phase with a lot of patients. So until this moment, it needs to be known if this drug inhibits one of the most famous transporters. And it's the same for substrate studies. In general, it should be performed until phase 2, but latest on before starting phase 3 clinical studies. So it shows also here the early discovery, maybe for bilirubin secretion, or for drug and metabolism DDI studies, also in the preclinical. In the phase 1 should be known something about the target tissue and the route of the drug, and you have, of course, performed animal studies, so you will see where the drug accumulates, so you can treat or check the tissue. For example, if there are liver accumulation, you of course need to assess the uptake by liver transporters. And to show the importance of these interactions of drugs, I will show you this list. For example, here are shown different transporters. Most of them are kidney transporters, and also interplay with metabolizing enzymes. And there's a drug which is a perpetrator. That means this drug inhibits the transport or metabolism of the victim, another drug. And in the end, the AUC, the concentration in the blood, can be increased or decreased. The best example which I have in my mind is cerebrastatin and gamfibrocil. Gamfibrocil is an inhibitor of OATPs, and it inhibits CYP enzymes. And cerebrastatin needs to be taken up into the liver and get metabolized in the liver by CYP enzymes. So if you apply both drugs at the same time, the AUC of cerebrastatin will be increased, so to a toxic concentration. And what happened? That there was a withdrawal of cerebrastatin from the market, which is a big loss for a pharma company. They invest a lot of money and then there's a withdrawal. So this should not happen. That's why such drug-drug interactions needs to be studied. And this is not the only key. Another key is that it's still increasing, that in the US population, more than 50% until now of the patients takes minimum 1 drug. More than 30% minimum 2 drugs, and more than 10% minimum 5 drugs. So that shows the importance of assessing drug-drug interactions. And as older the population is, here young population, most time 1 drug, less 2 or more drugs. And as older the population, of course, in the very older population, most of the patients take 5 or more drugs. So the risk for drug-drug interactions is really high. And what's really often observed in older patients is, for example, that the liver, or not the liver, that the kidney will be not anymore that functional. And this based most time on drug-drug interactions. So it's very slow developing such drug-drug interactions. And therefore, where the FDA made the first guidance in 2006, mentioned that it would be nice to study PGP. Then in 2007, there was a formation of some leaders or some people which know a lot about transporters. And 3 years later, they published their ITC white paper, where they already included that PGP is that it's essential to be assessed. Each new drug, the effect on PGP, if it's a substrate or an inhibitor, and they recommended also BCRP, OATP1B1, 1B3, OCT2, OAT1, and OAT3. The next step was that in 2010, the European Medicine Agency published a draft guidance and included already all these transporters as essential. They need to be studied. And they mention also, or they recommend, BCP and OCT1 should also be studied. Shortly after this, 2 years later, the FDA revised their draft guidance and they published a new draft guidance where also all these transporters which are here as essential are also here essential to be studied, and they recommend BCP but not OCT1. They also already mentioned that MATs and MRPs should be studied. It depends on the tissue where your drug will be delivered. Then in 2013, the ITC published much more transporters, so they went more detail how to study the transporters. And then, based on this, the EMA made a final guidance, more detailed, but as I said, they already wrote just the indicated transporter should be assessed. They never say directly this transporter or this, so if there is a liver accumulation, they just say the indicated transporter should be used. That means, of course, OATPs, but OCTN and NTCP, for example, can also be assessed. And I need to mention, we have a lot of transporters in our body, more than 400 transporters. So maybe the target of the drug is another transporter which is not the main transporter, so makes it a little bit difficult. Also, the Japanese guidance also published in 2014 their guidance, which is something between the EMA guidance and the FDA guidance. So I don't want to go for this in so much in detail. And now the recent one is the published draft guidance from the FDA in 2017 in October. And here they included the MATE transporters, and they still write or recommend BCP and MRPs. It was originally planned by the EMA also in 2017 to publish a new guidance, but the new time frame will be maybe in 2019, with much more details. I will talk a little bit more about the recent FDA guideline. This guidance is separated in two parts, in an in vitro and an in vivo part. So in the in vitro part, they go more details on the CYP enzymes and on transporters and how to study the transporters. They, as I mentioned, MAT1 and MAT2K are already included. And that's very important, I think, the most important part of this new guidance is the non-specific binding should be really assessed. So also in this, for these in vitro models, the drug can attach to the plastic material, can attach to the cell membrane. So the non-specific binding to all this needs to be included to come to the real results of substrate concentration and of the IC50 value. And this is something which we also right now spend a lot of time in how to set up or how to make these assays ideal or optimized for this, for including this non-specific binding. In addition, they also recommend to perform a 30 minutes pre-incubation with OATP 1v1 and 1v3. If you go on our webpage and you register, you will find our newsletter. And we published shortly after this, I think 2 weeks later, a newsletter because we did already time-dependent incubations. And with 50 minutes, 30, 45, 60, up to 90 minutes, or I think up to 3 hours. We observed the best inhibitory capacity at 90 minutes before, but they recommend 30 minutes. So just if you want to see more details, you can find the newsletter. And of course, the assay validation. Rollo mentioned already that from company to company, each cell line is different. And they recommend that each company should characterize each cell line with different substrates and test it with different inhibitors, so that each transporter overexpressing cell line is optimally characterized before they then supply their characterizations with their drug, because there are so huge variations. And I just want to mention, all of our cell lines are well characterized with different substrates and different inhibitors. The EMA guideline, which they want to publish maybe, hopefully, in 2019, they want to spend also on the recommendations for the transporters, maybe new transporters, maybe not, we don't know. Also on the time-dependent inhibition, and also, which is until now not really focused on the induction of transporters. I mean, they want to update this list. Also, they want to use other cut-offs for transporter inhibitions. This is a limiting step, also the uptake into, for example, liver cells by transporters. It's a limiting step. So they want to focus also more on this. Maybe they will say not anymore the indicated transporter, they will maybe more detailed. And with DDIs for contraceptive steroids. Maybe I wrote somewhere that's also important, or they will also mention something about SNPs, so gene variations. Sometimes OATPs or from their gene variation from patient to patient. So this is, I think, also something which will come soon in one of these guidances. So already included in the guidances from the European Medicine Agency, FDA, and the Japanese guidance are PGP, BCRP, as essential to be assessed for inhibition studies. Also, for substrate studies by the FDA and the Japanese guidance, the EMA they just consider until now. So I think consider means for me it should be tested. It's not a question. It's not a no. It's like a yes. Then we have other efflux transporters like BCP or MAPs. The EMA says for MAPs no. The FDA consider, which means yes. I think if you know the target and, for example, if there are liver efflux transporters involved, and you check, for example, PGP, BCRP, and BCP, and you see that it's not a substrate, of course you need to go ahead with the other efflux transporters like MRP 2, 3, 4. Okay, then let us go ahead. Of course, the hepatic uptake transporters, OATP 1v1 and 1v3, they need to be studied, the inhibition of drugs on these transporters. If there is a hepatic elimination, also the substrate studies need to be performed for all three guidances. Also another hepatic uptake transporter, it's the same, excepting the EMA just considered. For kidney transporters like OAT1 and OAT3, and also OCT1, excuse me, I mixed up. OAT1, OAT3 and OCT2, they are kidney transporters, though the inhibition on these transporters needs to be assessed, that's essential. And if there is a renal elimination, also substrate studies need to be performed. So what means inhibition studies and what means substrate studies? So inhibition studies means the new drug should be used as perpetrator. So the new drug will be checked if it affects the transport of another drug. This is inhibition studies. For substrate studies, the drug will be a victim. That means have another drug an effect of the transport of the new drug in development. So these are the both types, and here I will show the first decision tree in general. All investigational drugs needs to be characterized for PGP and BCRP. So if they are substrates. If a drug shows, for example, in an animal model, hepatic or biliary secretion, which is higher than 25%, the uptake by OATP1 and OATP1B3 needs to be assessed. If there is a renal elimination, also the hepatic renal uptake transporters needs to be assessed. And I wrote it on the side because in the new guidance there's not such a nice decision tree. Also MAT1 and MAT2K needs to be assessed. Now I want to go a little bit more over this table to go more in detail the essays behind the decision trees. And now I want to start with the very essential both efflux transporters, PGP and BCRP. The EMA wrote that if there is as I already mentioned, if there is a drug elimination higher 25%, a biliary, also in the gut wall, the transporters involved needs to be studied. The FDA and the Japanese guidance, they already mentioned that it's in the gastrointestinal tract, both transporters, liver and kidney. And I also want to mention in the brain, in the blood-brain barrier, also both transporters are essential. So if you want to treat in the brain, you also need to be study both transporters. The decision tree, I really want to show. Where is it? 1 second. In our book, which we have, so we just have a few books with us. So you can also register on our webpage and you can order a book for free. We have made very well decision trees, so that all these decision trees for all these tissues, for all the transporters are pretty well described. So I just recommend, if you want to really study and if you want to understand how it looks, have a look at this book. It's very nice described. And for example, here it's shown for PGP, but it's also for BCRP the same. The FDA recommend to use this CACO2 cell monolayer or just MDR1 overexpressing cell lines. And if there's a net efflux ratio, Roloff already mentioned which is higher than 2, then it needs to be tested more in detail by giving an inhibitor, and can be the uptake or the efflux ratio inhibited, then it seems to be a substrate. If the efflux ratio is below 2, that means it's maybe not a substrate. So it's the same for BCRP. And the decision tree is, you will see it, it's most time for all the transporters closely related. Here I want to show, this is the decision tree as it looks like in the guidance. So I think we made very nice, so it looks just, it's easier. Here's, I mentioned right for inhibitor, for substrates, and here's for inhibitors. That means it's also for CACO2 cells or MDR1 overexpressing cell lines. And if the efflux ratio of a probe substrate gets inhibited by the new drug, then it's probably PGP inhibitor. Then needs to be performed KI or IC50s. So IC50 is recommended by, for example, the FDI, the European Medicine Agency. They want to have KI study. If I missed the word. The Cmax divided by IC50, is it higher than 0.1? Or if the dose divided by the IC50 higher than 10, then it looks like an inhibitor. If it's not, if it's below 0.1, below 10, then it's maybe not an inhibitor with a high probability. So that means I want to go to the CACO2, but already Roloff talked about it. It's a very good method, especially for PGP and BCRP. It studies a lot of transporters, all transporters which are expressed. It's widely used. It's also recommended by the guidance, so I recommend to use this cell line to get a good overview. If you want to study a transporter in detail, we recommend to use the vesicular transport assays. They are already accepted by the guidances. It's quick, easy, and also cheap. And of course, CACO2 cells, they need about 3 weeks to get tight, so in the wells. So vesicular transport assay, you perform it 2 hours later, you have the result. So that's a huge difference. A very good point for CACO2 cells is, I think, you have one cell line and you can study directly 3 efflux transporters. So if there's an efflux ratio, you observe an efflux ratio, you can block using specific transporters, for example, PGP. Then you can say, okay, it's maybe a substrate of PGP. You can block BCRP. Then you have maybe can say something, if the efflux ratio afterwards decrease, it's maybe a substrate of BCRP or of MRP2, for example. So for this, the cell line is quite a good opportunity. Let's go ahead. Now I want to focus on BSEP and MRP2s, and they are just considered or not considered, and that's why I just want to give a short overview. The EMA, they just write that they prefer assessment of BSEP. So I think if an in vivo model, for example a mouse, shows that there may be an effect in the biliary secretion, then of course BSEP needs to be characterized. Also the FDA, they just consider. I think this really based on in vivo observations. And the ITC, they recommend if there is a liver toxicity, it's better to check maybe there is an accumulation of the bile acids. For MRPs, the EMA until now they didn't mention it. Maybe in the next guidance, the FDA, they just consider, as I mentioned, if you observe, if you know there's a secretion, and none of the classical three efflux transporters, BCP, BCRP, and PGP, are the transporters involved. Of course, the MRPs needs to be studied. The ITC recommend to assess MRP2, MRP3, and MRP4 in case of hyperbiliruria and glucuronidation observed for the test article. For OATP1B1 and OATP1B3, they are very important, and I am still wondering why they never included OATP2B1, because I think both it's an overlapping substrate spectrum. But it can also be happen that your drug is just a substrate for OATP2B1. So don't be too focused on just both transporters. Also think about that there is also another uptake transporter, which is also expressed a lot in the cardiovascular system, I need to mention. Here the EMA, FDA, and also Japanese guidance says if there is a hepatic elimination, it's of course important to check if the drug is a substrate of these transporters. And here for overexpressing cell lines needs to be used. The decision tree, it's again the same. If the hepatic elimination is higher, 25%, if there is an accumulation in the liver, also under physiological properties, of course it needs to be checked if the investigational drug is a substrate. And this is very important for this is for substrate assessment. I wrote it this morning, this is wrong there, for inhibition studies. 30 minutes pre-incubation is right now recommended by the FDA. So, very important. So, if the uptake of the substrate of the OATPs can be inhibited, so it looks like an inhibitor of the transporters. And of course, as I mentioned, both transporters needs to be assessed right now with the 30 minutes pre-incubation for inhibition studies. And let's go to the kidney transporters. The EMA, they just say again about this 25% of the drug. If there's a renal elimination of higher 25%, renal transporters needs to be assessed. And they just write the indicated transporter should be used, so they don't focus. So for the EMA means already also the MATEs should be tested. The FDA right now say it more clearly, OAT1, OAT3, OCT2, MAT1 and MAT2K, they needs to be accessed for active uptake. Also the ITC, beside MATs, they also mentioned more to use OCT2. And the PMDA, they already mentioned all of them. So the FDA and PMDA, they are already at the same level right now. And the decision tree is again more or less the same for substrate assays. The uptake needs to be assessed for all these transporters, and if there's an accumulation greater than empty vectors, that means an accumulation of a significant accumulation, then it's likely a substrate. And can be, and if this substrate uptake can be inhibited by an inhibitor, then it's likely really a substrate. And for inhibition studies, again, it's always the same. The transporters, uptake transporters of the kidney, different concentrations of the compound needs to be assessed. for determination of IC50s. So, of course, then there's another step, the unbound Cmax versus IC50. If it's higher of 0.1, it looks like a substrate, the drug. And here I just want to switch back quickly to the hepatocyte system, because you have in the sandwich culture and also in the hepatopac system, you can do all of them, all of these liver assessments in just one type of assay. So I just wanted to mention this at the end, that it's already possible. They are very complex. You can perform it in all of these in one assay. It's also, I don't have it for the slide for the proximal tubal cells for the kidney. It's also possible to do all these steps in just one cell type or one type of assay. Yes, here I also, that's why I added just this slide. The proximal tubal cells can be also used because they have the full spectrum. Using specific inhibitors, you can really study it as one step. So, and that's it. Thank you.
Thank you very much for your presentation. Any questions? I think we still have some time. No any questions? Yeah, I think they present very thoroughly, very clear about their platform, which actually can help us to do very comprehensive study in terms of in vitro assessment of the transporter, for the DDI potentiality of your compound assessment. I think that is very, very clear, very good presentation today. And if you are interested more, I would suggest and recommend the colleague to refer to 2017 draft guidance of the US FDA. They also have a very clear guidance, although it's not very long, it's very short for one. Just not very long guidance, and you get a lot of guidance in terms of strategy and study design, and also the technical aspect. So I think this issue, this topic, actually investment getting a lot of attention from the regulatory agency. So that's why they come up more clear guidance on that. 2012, US FDA already have a guidance. Now they want to split into two. One guidance is in vitro, specifically focused on the in vitro. Another one is specifically focused on the clinical DDI study. And these two guidance is going to replace their previous 2012 guidance. And that's been the agency now have more idea about how to regulate in this area. And I think specifically what in vitro part, so the platform seems to be a very good one, a very comprehensive one. They have a lot of information provided to us today. You can contact them or talk to them later if you are interested. Anyway, we thank you. Thank you so much to Nora and her team, Daniel and Rodolfo. And very comprehensive, very wonderful presentation. Thank you. And thank you all your attention and your attendance. Thank you very much.
I just wanted to mention, please don't hesitate. If you have any question, write a mail and we will answer.